This is a scanner at Hammersmith Hospital. It's actually a 1.5T MR scanner, which we can use to carry out something called multinuclear spectroscopy. We can take someone and scan their brain and not just look at the structure of the brain, but also get a handle on the chemistry of the brain, actually find out about the chemistry in real time in the brain, totally non-invasively. It's absolutely safe. There are no adverse side effects, no injections, no ionizing radiation. Very powerful technique. I'm afraid the ugly mug there is yours truly. I haven't got a picture without me on, unfortunately. We carried out the first systematic study of the chemistry of the brain in ME, and what we found was that the patients with ME had a significantly raised level of choline-containing compounds. Choline was also mentioned, I think, by Malcolm this morning. In fact, I went back over the original scan data recently, and every single ME patient had a level of this peak which was higher than that in every single normal control in our study. Well, what's this mean? Well, the first thing you have to do when you obtain a result as dramatic and as striking as this is to see if it's reproducible. And in fact, a year later, Professor Chowdhury's group published a similar study, same protocol, same sort of scanner, same numbers of patients and age and sex matched normal controls, and their findings were the same. And Ray Chowdhury pointed out to me that in the year 2000, in Japan, Tomodo and colleagues had published not a systematic study, but a case series of 3 young ladies with juvenile ME, and they'd had this particular investigation, proton neurospectroscopy, and they had shown exactly the same as well. The only finding was clear raised level of choline. So the issues I'm going to address today are, first, what does this striking result mean? And second, what, if any, are the implications for treatment? So what does it mean? We have to go back to the cell. As you all know, our bodies are made of organs, our organs are made of tissues, and our tissues are made of cells. Now, each cell is bound by a lipid membrane, and inside each living cell are organelles, such as the nucleus, the mitochondria, the powerhouses of the cell, and so on. And many of these are also bounded by lipid membranes. And if you study them carefully, you find that these lipid membranes, which are terribly important for processes such as neurotransmission, the transmission of signals from one cell to another, these lipid membranes have 2 layers. And if you drive down deeper, you find that key to the structure and, in fact, to the functioning of these lipid membranes are these things. They're called phospholipid molecules. And you can see there are 3 components, these spherical type things, and then these squiggly things coming off. And actually, one of them is squiggly, one of them is a little less squiggly. The spherical things are parts of the phospholipid molecule that like water, and they are things like choline. Remember the choline that we found, which was raised. And the squiggly things are fatty acids. It turns out that if you don't have the right fatty acids in your body, then you can't make these phospholipid molecules. And so you have too much of this choline, or whatever other hydrophilic group it is, lying around free in the brain. And that's, of course, what I measured using proton neurospectroscopy all those years ago in ME patients. So is there any reason why in ME there aren't sufficient levels of those right fatty acids? Is there any evidence for that? Is there any reason that could happen? Well, here's just a different projection of the same thing. This is called a Fisher projection. It's exactly the same thing. And we're concentrating on that particular fatty acid there, do we have enough of those? The key long-chain omega-6 and omega-3 fatty acids we need are arachidonic acid, DGLA or dihomogamma-linolenic acid, and eicosapentaenoic acid, or EPA. This is the busiest slide I shall show. And you'll see from this, if you look carefully, here are the omega-6s, and those are the omega-3s, that from our diets, we very easily obtain very large amounts of LA and ALA. These are the precursor starting fatty acids. And then each of our living cells has these enzymes, by means of which it can take each of those fatty acids and do things to them, elongate them, introduce fatty double bonds and so on, until you end up step by step with the key fatty acids we need. And in particular, as I mentioned, DGLA and AA on the omega-6 side, and EPA, and to a much lesser extent, DHA on the omega-3 side. So there should be no problem. I've just said we have ample amounts of LA and ALA in our diets, far too much probably of LA these days, and we have the chemical machinery by means of which we can convert those precursors into the longer chain omega-3s and omega-6s. Whence the problem? The problem arises right at the beginning there, with delta-6 desaturase, and perhaps I could try to explain to you why this problem arises. I've shown you the structure of phospholipids, and you've seen that arachidonic acid and one of the long-chain omega-3s are important components of those. But 3 of these fatty acids have other extremely important functions besides structure. So DGLA, AA, and EPA are also the starting points, the precursors of all the eicosanoids that we need. Eicosanoids, long name, what does it mean? It refers to whole families of terribly important substances which are absolutely vital to maintain the health and well-being of all our tissues. All our prostaglandins, all our thromboxanes, our leukotrienes, our interleukins, our cytokines, our hydroxy fatty acids come from DGLA, AA, and EPA. And amongst other things, they play a vital role in defending our bodies against infection, and indeed also in enabling our immune systems correctly to identify cells that are beginning to show DNA mutations and allowing our immune systems, as it were, to zap those potential foci of cancer. So they are really absolutely vital. And it is important if a virus is going to try to establish itself in our bodies that it stops our bodies making EPA, DGLA, and AA, because these 3 fatty acids are the way in which our immune systems function properly. They are the way in which our immune systems obtain all the key chemicals that are needed to defend ourselves against those bacteria. And it's more reasons beside. EPA itself is directly virucidal. That is to say, it kills viruses better than domestos. Really does. I mean, if you would take a blood sample from me, now that I'm back on my fatty acids, and you were to add that to some of the more lethal viruses, my blood would kill those viruses. I haven't done it in my blood. I don't need to. The papers are out there showing that physiological concentrations of EPA are directly virucidal. Also, we've heard a lot about interferon beta just now from Jonathan. EPA gets converted by a couple of enzyme systems, cyclooxygenase and lipooxygenase, by the body into interferons. Jonathan mentioned that interferon is, or the interferon family of proteins are endogenous. They occur naturally in our bodies. Well, if you take a lot of EPA, the EPA, some of that gets converted into interferons, and they, of course, are virucidal as well. So by 2 different mechanisms, a 2-pronged attack, EPA kills viruses. So if you are a virus, and I know I'm speaking teleologically, but if you were a virus and you were trying to establish yourself in the body, you should zap this system. You should inhibit it from working. And that's what viruses do. We know from in vitro experiments that they inhibit very strongly this enzyme here, delta-6 desaturase. They stop the whole of our body's machinery from making these long-chain fatty acids from working in the first place. There are other inhibitory factors as well. Stress is terribly important. And the way that stress acts, and stress isn't a very good term, as medical students were taught not to use the word stress, but I think you know what I mean by stress. Stress operates because, as an inhibitory factor, because it causes the cortex, the outer part of the adrenal or suprarenal glands, which sit just above the kidneys, to secrete large amounts of cortisol, stress hormone. And being a hormone, it's circulated throughout the body, in our arteries and capillaries, and it reaches all our cells, and it inhibits delta-6 desaturase from working. A deficiency of certain key micronutrients is also now known to inhibit delta-6 desaturase, as is excess intake, perhaps even small intake actually, of nicotine and caffeine. I hope you enjoyed your tea, by the way. So, is there any evidence that viruses are implicated in ME? Well, of course, the answer is yes. You've seen a lot of evidence over the course of today, and I'll just summarise a bit of it very quickly. We know from some of the key epidemics, like the Los Angeles County Hospital epidemic in the 1930s and the 1950s royal flea epidemic, that these epidemics do seem to have the clinical appearance of a viral infection with phenomena such as cervical lymphadenopathy, enlargement of lymph nodes in the neck, and changes in muscle structure and function, and even enlargement of the liver, hepatomegaly, occurring in people affected by those epidemics. Another bit of evidence, of course, relates to the immune system. I'll just flash this by very quickly to give you a flavour of just how complicated our immune system is. But every time you see these pictures, when you see certain molecules, like those, there we are, gamma interferon or interferon gamma and various interleukins, just bear in mind, flag up in your mind that those key parts of our immune systems are derived from DGLA, AA, and EPA, those three key long-chain fatty acids. And this is an interesting study. There was a meta-analysis, a large study of immune system functioning, published a couple of years ago. I think, if I remember, the first name of the second author was Simon something I can't remember. I'm sure you can think. And this was very interesting because they found this particular pattern of changes. And of course, we know what this pattern of changes means. It's entirely consistent with a persistent viral infection. I mean, it's also consistent with an autoimmune disorder where you attack your own body, but there's no real evidence that autoimmune diseases are more common in ME than in the normal non-ME age or sex-matched population. And then there's been limited work on fatty acids in ME, and they've also found evidence of a reduction in key long-chain fatty acids. And of course, there's the work that I mentioned at the beginning by our group at the Hammersmith and by Chaudhry's group at that time at Southern General Hospital in Glasgow, and also from Japan. And those are the three studies I know of. And there's been Jonathan's excellent work recently, which also, part of which also is consistent with a persistent viral infection being present. The idea that someone can succumb to a viral infection, be ill for a week or two with terrible symptoms, flu-like symptoms and worse, and then apparently no longer have the virus in their body, but still be subclinically infected, that notion is well known to those of us who practice medicine, but it may not be quite so well known to those outside of medicine. So I thought I'd just remind you of two good examples, which I'm sure you're all familiar. Here's one, the common cold sore, caused usually by herpes simplex virus type 1 or type 2, HSV1 or HSV2. And someone will catch this, and it will eventually go, hopefully. But just because the cold sore disappears doesn't mean the virus has necessarily disappeared from the body. On the contrary, the virus can migrate up one or other of the relevant nerves, right up to the trigeminal ganglion, and then remain there and reproduce there, but as a latent infection. And the person doesn't realise they've still got the HSV1 or HSV2 viral infection. And then years later, when they are subjected to physiological stress of some form or psychological stress, the viruses can come back down and cause the cold sores again. And here's another example, chickenpox. I'm sure it is adult chickenpox and affecting someone's face, and here it is affecting someone's trunk. And these skin lesions can disappear, but just because the lesions disappear doesn't mean that the causative virus or viruses have disappeared. On the contrary, they can migrate again to one of the ganglia inside the head or one of the ganglia on either side of the spine, and then years later, they can, when they've been reproducing and establish itself in the body and causing all sorts of problems, but at a subclinical level, they can again manifest themselves as an infection. And of course, it comes back as, I'm sure you'll recognize it, shingles, yeah. In the thorax, and here's shingles affecting. You see, it just affects one side, that you can tell which ganglion it went to, because it's from the anatomical distribution of the shingles when it comes back. So we can have these viruses inside us. They're very clever, really, I mean, from an evolutionary point of view. I'll just check the time. Okay. So I'll skip through these to save time. I've mentioned viruses many times, but we shouldn't forget that there are other organisms that can infect humans, and in particular, there's a spirochete that can do it. And so second on my list of candidates is a spirochete, and I'm particularly indebted to Jane Bryant and Angela Kennedy, who I think have done a lot to bring British public awareness to the fore about this possibility. I mean, in America, I think people are more familiar with this potential infection, partly because, of course, it was first discovered as an infection in humans in Connecticut, a tick-borne infection caused by this particular spirochete. In fact, it's named after the discoverer, William Burgdorferi, in 1983, so it was christened in 1984. Right, so these are the fatty acids we need, some of them, and there have been some fatty acid trials. I'll go through these very quickly. The first was published in the early 1990s. It was a 3-month trial using fatty acids that were given to patients in Scotland, and again in Glasgow, in fact, and there were positive results seen as early as 1 month, and certainly highly significant differences between two groups by 3 months. Since then, there haven't really been any very large-scale studies using high or preferably pure EPA preparations with evening primrose oil. You might wonder why we've got this combination here. Well, the EPA, obviously, we need EPA for the omega-3 side, and the virgin evening primrose oil is an excellent source of GLA, which gets converted very easily to DGLA and AA. So you get your DGLA and AA on the omega-6 side and your EPA on the omega-3 side. But what studies there have been have, again, indicated benefits. There have also been benefits we found in respect of brain structure, and actually in respect to the amount of brain tissue available. And just yesterday, my latest paper on this came out. It's this one here. I think one of the people in the audience has several copies of it and wants one later. Beware, the title's one sentence, is 40 words long, the titles. Are there any other factors that can contribute to a deficiency of fatty acids? Well, there are many, actually, besides viruses. Our modern diets are, unfortunately, rich in junk food, in inverted commas, and also our modern diets cause us to have a very poor intake of vitamins and trace elements. I say us, I don't include myself actually, but most people. And our diets, people's diets, are rich in trans fats in Britain at any rate. I just don't know why, when the United States of America has now agreed that manufacturers have to state the levels of trans fats in products sold in supermarkets there, when Denmark has, I think, forbidden the use of trans fats beyond a very, very low level in foods sold there. Why we have no such legislation in Britain? I mean, you know, why should we carry on suffering in Britain? I have no idea. And there are lifestyle causes as well for deficiency fatty acids. Finally, I'm just going to go briefly through the protocol I currently use with patients. It's very logical, really, and really quite simple, I think, really. First, I exclude all other diseases, and we heard a lot about this earlier on today. Then I seek actively to bypass the block at delta-6 desaturase. Third, I try to ensure that my patients get sufficient levels of vitamin and mineral cofactors. And fourth, I have no problems at all, even though I'm steeped in traditional medicine, I have no problems at all in recommending those complementary therapies which can help with alleviating stress and encouraging lymphatic function when there is evidence to support those complementary therapies. So let's just go through those one by one quickly. Exclusion of other diseases. I too carry out a very full medical history on my patients. It doesn't take me quite 1.5 days, but it does take a long time. A full mental state examination, an absolutely very thorough physical examination. It really is unbelievable, really, the number of patients who are referred to me, some of whom have come via the private sector and paid hundreds and hundreds, sometimes thousands of pounds to people in the private sector. They come and see me, and they've never had their thyroid glands palpated properly, they've felt properly, they've never had their chests examined properly, their abdomen examined properly. The woman may never have had a breast examination, they've never had a proper central nervous system examination, a peripheral nervous system examination. I just don't understand it, but I do, you know, I'm of the old school. I do actually believe that it's rather helpful to examine one's patients. And then we try to carry out a whole range of investigations. The most invasive of those investigations is simply taking a blood test, and all the rest are non-invasive. My wish would be to carry all those out under a research protocol, but alas, recently we failed to get ethical approval to do just that. I'm not sure why, so we'll be reapplying again. There's Megan, who'll be reapplying again, 30th of May. The thing's crazy, really. They changed the COREC application for medical ethics on the 1st of May 2004. Sorry, this is. I wasn't going to talk about it, but I think we have an MP here. Yes, I'll just. Oh, he's gone. Oh, in that case, I won't bother. Okay. So I choose to bypass the block at delta-6 desaturase, and I do that using ultra-pure EPA with virgin, even primrose oil. Be very careful not to include DHA in your supplement. The body will make the DHA itself from EPA. We also need cofactors. In particular, we need certain vitamins to make this whole system work properly. Biotin, niacin, folic acid, vitamin B6, B12 and vitamin C or ascorbic acid. We also need certain minerals, in particular zinc, magnesium and selenium. One possibility is to give your patients a cofactor supplement, something like Vegeco, for example. But, of course, nature provides all these cofactors in foods, and if you can possibly manage it, you should try to encourage your patients to eat more fruit and fresh vegetables. But, of course, it's very hard, as someone called Fiona Harker was reminding me this morning, at lunchtime. It's very hard if you've got ME to sit there chopping your vegetables and cutting your fruit and peeling your potatoes, etc. You shouldn't peel your potatoes. You should boil them completely. There's lots of nutrients just under the skins. And so there are alternatives, and I'm grateful to Fiona for introducing me to the fact there is a supplement called Juice Plus, which just contains concentrated levels of fruit and vegetables and nothing else. There are lots of patients who have joint problems, and it turns out from studies of osteoarthritis and rheumatoid arthritis in non-ME patients that the key biochemical change in the synovial fluid, which bathes the joints, is a lack of EPA in there. So obviously if you take a supplement that's very rich in EPA, you can help reverse that. EPA also gets converted into natural sleep mediators. It doesn't just get converted into interferons, it gets converted into natural sleep mediators. So sleep should improve enormously, but you do need an extra cofactor. I forgot to mention it. And then on the complementary therapy techniques, one that I favour a lot is the Alexander technique, but it hasn't actually been systematically applied to ME, sadly. But it does, it's very, in fact, making me want to stand up straight and adopt a better posture. And there's one other complementary technique which has been tested thoroughly in ME. It's the what I call the Perrin technique, and I think the gentleman's here himself, the man himself, Dr. Raymond Perrin, and he wrote a whole thesis on it, which this is just a tiny summary of it, for which he was awarded a PhD by the University of Salford, I believe. So finally, my conclusion, obviously, is that we need more research, but I'd like, in particular, to thank the MRC, Hammersmith Hospital, Imperial College, Merge, and ME Solutions. Thank you very much.